And I'm Xihong Lin. I'm a professor in the Department of Biostatistics and Statistics at Harvard University. I would like to thank the conference organizer for inviting me and give the presentation. And today I'll share the work on the analysis of large biobank and multi-ethnic whole genome sequencing study. So this is the timeline of the whole genome sequencing in the last 20 years. As the Human Genome Project was finished about 20 years ago, and about 12 or 13 years ago, that was the launch of 1000 Genome Project. At that time, because the sequencing cost was quite high, so therefore only about 1,000 people were sequenced, and also the depth was pretty low. And in the last few years, the sequencing cost has dropped dramatically, and so the much more number of people have been sequenced. And there are two large whole genome sequencing project going on in US, and one is called Transomic Precision Medicine Program by NHLBI, that is also called TOPMed, and so it sequenced about 200,000 people. And the other is called the Genome Sequencing Program by NHGRI, and they sequence about 150,000 whole genome and another 200,000 whole exome. And the NCI started its own sequencing program, and so it sequenced both the germline and also somatic genome. And then there have been a large number of biobanks which have emerged in the last few years, and in the most well-known one is the UK Biobank. And then in the next few years, we are going to see tens of millions of whole genomes sequenced data available, and we need to be ready for analyzing such large data. So here is a quick overview of the NHGRI genome sequencing program. It's multi-ethnic. It has about 140,000 whole genome and also about 200,000 whole exome, and it has 4 complex disease genomic centers and 4 Mendelian disease genomic centers, and also 3 analysis centers. And we are one of the 3 analysis centers. So here's a quick overview of the TOPMed. And so the TOPMed has about 190,000 samples, and it's also multi-ethnic, as you can see in the pie chart on the left. And they are about 40% Europeans, 30% African American, 15% Latino, and about 9% Asian. And also it has many phenotypes, and including lung and heart phenotypes, and also cohort including multiple phenotypes, such as the Framingham study and the Women's Health Initiative, and also the blood phenotype and sleep phenotype as well. And besides the whole genome sequencing data generated by TOPMed, TOPMed also generated the whole multiple omic data, including RNA-seq data, DNA methylation data, and proteinomic data of a subset of the samples. So here is the frequencies of the TOPMed Freeze 9 data. So as you can see that in TOPMed Freeze 9 data, there are almost about 700 million variants, and a large fraction of them are rare variants, and so about 97% are non-coding variants, and about 46% are singletons, 16% doubletons, and you can see there are only about 1% are common variants. So what that tells us is that there are massive rare variants in the whole genome sequencing data, and unlike the common variants analysis, and which is done in the GWAS, and each variance can be analyzed separately, when we deal with massive whole genome sequencing data with massive rare variants, and we cannot do individual SNP analysis because of lack of power, so therefore one need to do SNP set analysis. And the biobanks are the trend in health science, and biobank nicely integrate genome data with electronic medical record and epidemiological data, imaging data, and wearable devices. And so here are a few examples of biobank, including the UK biobank, which has 500,000 people, and it has the GWAS data, which is already public available. And right now, it has the whole exome sequencing data of 300,000 samples. And by the end of the year, will be 500,000 sample with whole exome sequencing data available to the research community. And by 2023, whole genome sequencing data will be available and for the UK biobank. And also China Kadoorie and Finland biobank, Million Veteran Program, Geisinger, eMERGE, and many others. And RVAS is another good example. It used to be called the Precision Medicine Program and announced by Obama administration, and then they tried to change it to called RVAS. And it also multi-ethnic, and is the plan is to enroll about 1 million subjects and follow them over time. So the goal of those biobanks are to help with the precision health, including precision prevention and precision medicine. So in the next few years, we are going to have millions or tens of millions whole genome or whole exome sequencing data available. And so we need to develop a scalable and tools to analyze such large data. So the talk has 3 parts. The first part I will present the RVATS association test in whole genome sequencing study, in particular the STAAR method, by adaptively incorporating multi-dimensional functional annotation. So basically, by empower RVATS association test, upweighting the functional variance using multi-dimensional functional annotation scores. And so this work was led by my postdoc Xihong Li and Zilin Li, and was published in Nature last summer. So let's start why we want to integrate the whole genome sequencing data with the multi-omic data. So the one advantage of doing that is by incorporating multi-omic variance functional annotation data, and then into the whole genome sequencing data, and one can empower RVATS association discovery by upweighting the functional variance, and also by creating the meaningful masks. And secondly, by incorporating multi-omic data, it can also help understanding the causal pathway from the variance to function mechanism to phenotype. For example, this can be done through the causal mediation analysis. And then the ultimate goal is transform the knowledge into action, and with those genetic discovery and help with the drug target identification and prevention intervention strategies. So today we are going to focus on incorporating multi-omic data with whole genome sequencing data to empower RVATS discovery. To do this, we first need to do functional annotation of the whole genome sequencing data. And to this end, the NHGRI genome sequencing program launched a few years ago, Annotation Working Group. And so the goal of this working group is to develop a comprehensive whole genome functional annotation database to facilitate result interpretation and also whole genome sequencing analysis. So we integrate the data from many different omic sources, including ENCODE, Roadmap, ANOVA, and CAT, GTAG, and Phantom, and so on, into this we call the FAVOR database. And so currently, this database has over 300 non-tissue specific annotation and also tissue specific annotation. It's 80% of the annotation were built on the H38. And by doing that, that can provide multidimensional functional annotation, including allele frequency, conservation score, protein functional score, epigenetics, and so on. And also, so right now the FAVOR database is available online, which has functional annotation of 500 million TOPMed variants. And so we are currently working on providing the functional annotation in the FAVOR backend database and also online portal with 3 billion positions. And so then one can use that to query the functionality of any variants, or the genes, the variants in the genes, and or the regions. And also one can use this backend database to annotate any whole genome or whole exome sequencing study, such as UK Biobank. So this work was led by my computational biologist, Hufeng Zhou, and also software engineer, and Ted Procola. And because there are many functional annotations, so we first need to do dimension reduction in order to incorporate such functional annotation in the whole genome sequencing real-world analysis. And to this end, here it shows the heat map of the functional score and across the different domains. And so, for example, so for epigenetics, there are probably 30 different scores, as you can see in this big block, and they are correlated, and also the conservations, and they have good correlation as well. And so therefore we want to do the dimension reduction. And so for each categories, and we create the annotation PCs. And so this is different from the ancestry PC. Ancestry PCs are calculated for each subject to control for population structure. Here, the annotation PCs are calculated and for variants, and those measure the functionality in multi-dimensions of each variance. So therefore, we can see for each variance, we calculate annotation PCs, and for epigenetics, and basically this is the first principal component of those 30 epigenetic scores, and also annotation PC for conservation, annotation PC for protein function, local diversity, and so on. And then we use those as a weight in the real-world analysis. Then how can we do that? And so before we do that, we first need to resolve the data storage issue. Because the data are very large, we have to develop efficient algorithm or efficient databases to store the data effectively. So if you look at the example here, so for the CCDG Freeze2 data, which has a 60,000 sample, and then you can see the VCF file is large, is 250 terabytes, and if you use the BCF file, this is 2.8 terabytes, and then that the compression rate is 1%. If one use the GDS file, you can see the compression rate is 1,000 times. It's much, much better, much smaller. And then if one incorporate functional annotations, and then you can see that compression rate is still about 1,000 times. And so similarly for TOPMed data. So this will dramatically reduce the storage cost for large whole genome sequencing data. And now, what is the whole genome sequencing analysis flow? And so the input data include the phenotype data, the covariates such as ancestry PC, and also the GRM was a sparse GRM, which account for relatedness, and also the genotype data and functional annotation from AGDS file. And then one can perform two analyses. One is a common variance analysis, and by analyzing one SNP at a time. The other is to do the real variance analysis by analyzing a SNP set at a time. And for real variance analysis, one can do the gene-centric analysis by creating different coding masks and also different non-coding masks for each gene, and or the genetic region analysis using the sliding window approach with the fixed window size or dynamic window size we call the ScanG. So this is a quick illustration of the mask. So for gene-centric analysis, for each gene, we can create a different mask, such as loss functional variants and missense disruptive missense variants, synonymous variants, and for each of them create as one mask. And then for non-coding variants, and for each gene, we can group the variants into the promoter and promoter or enhancers and of each gene, or upstream or downstream. And also for the sliding window approach, one can use the fixed window size or dynamic window size. And so this basically is the STAAR analysis workflow. Then I'll talk about how to construct the statistic. So prepare the data, one can have the whole genome sequencing, genotype data, and then use that to create a sparse GRM matrix to account for relatedness. And also one can have the covariates such as the phenotype and also the covariates. And then one annotates the genome using the STAAR as a backend database, using the favor as a backend database. And so you can see that we annotate the variants of the, for example, TOPMed Freeze 8 data. And then for each variance, and we calculate epigenetic scores and or conservation score and so on. Then we calculate the first principal component of 30 epigenetic score to calculate APC epigenetics. Do the same thing for APC conservation and APC protein function. And so those are all available and in the favor data backend database. And then one creates the variant set. And for gene-centric analysis, one groups the variants into the missense variants or loss functional variants, promoter and enhancer for each gene. For non-gene-centric analysis, and one do the sliding window, either using the fixed window size or dynamic window size. And now one can construct the test statistics. So basically, one can use the different annotation as a weight. For example, if one uses the SCAT method, I'm going to explain in a few minutes. And then, so if you use minor allele frequency by upweighting the real variance at the weight, one can calculate SCAT p-value, or using the epigenetics APC as a weight, one can calculate SCAT p-value, or using the conservation as a weight, APC as a way to calculate SCAT p-value, and then we use the ACAT method to combine them. We can do the same thing using the burden method or using the ACAT-V method. Then afterwards, and we combine all of them using the ACAT method into the STAAR-O. And the reason one wants to do that is because the burden method works well, has a power when the causal variance, majority of the variants in a mass are causal variants, and also the effect are in the same direction. And the SCAT method allows the effect to be in different directions, and also allows us the variance not a fraction of the variants are non-causal. And the ACAT-V method allows sparse signals. And so therefore, STAAR-O method will be robust across all different scenarios. And then after that, then one create a p-value for each mass. For example, the association p-value of the loss functional variants of ApoE and LDL, and the promoters of the ApoE and LDL, and so on. And so, so now let me talk about how to construct the statistics. So there are two features in large biobank and whole genome sequencing study. And one is multi-ethnic. So for example, if you look at the figure on the left, you can see that for million veteran program, there are multiple ethnicities. And also in the TOPMed, there are multiple ethnicity. And also there are relatedness. So you can see that in UK biobank data, there are a good number of first degree, second degree, and third degree relatives. Similarly for TOPMed. So we want to take that into account in analysis. So how can we do that? So we do using the mixed effect model, using the linear or logistic mixed model for the continuous trait or binary trait. And so in here, the G will be the SNP in a mask. For example, 100 SNP in the promoter of ApoE, and X will be the covariates such as age, gender, and so on. And APC will be ancestry PCs. And B will be the random effect, and the covariance is sigma, which is ancestry adjusted sparse GRM. And then, so it's important to disentangle the ancestry PCs and from the random effect. Ancestry PC capture the population structure, and the random effect the ancestry adjusted sparse GLM capture the relatedness. We need both in the model. And to make the calculation scalable, so what we do is we fit the null model and basically the model without the genotype and only once. And then we calculate the score statistic for each SNP across the genome, and then we aggregate them within each mask. So how can we aggregate them? And so the traditional way, the SCAT method is, suppose we have 100 SNP in the promoter of ApoE, we calculate the score statistics of each SNP, square it, multiply by the weight, and sum over the 100 SNP. And then this works well when the signals are in different directions, and when signals are sparse, then ACAT method will be helpful. So basically, the idea is we calculate the p-value for each SNP and do a Cauchy transformation, and then add them up. And then this will be the ACAT method. So this is powerful, and when we have the sparse signals. ACAT method is also helpful when we use a different weight, because we don't know which weight works for a particular mask, because APC epigenetic may work for the one mask, but may not work for the other mask. So the idea is, suppose you have a different weight, and then how can we combine the different test statistics across the different weight? The ACAT method allows for us to doing that. So this is a beautiful method and was led by my former postdoc, Yaowu Liu. And so then this basically is the idea. Suppose we have, say, the promoters of the ApoE. It has 100 SNP. And then we use the minor allele frequencies as a weight. Then we calculate SCAT p-value, and then we do a Cauchy transformation. And then we use the CAT as a weight and calculate the SCAT p-value and do a Cauchy transformation. And or we use APC epigenetics as a weight and do the SCAT p-value and then calculate the Cauchy transformation and so on. Then we do, so you can see we use a different weight to calculate the SCAT p-value, and then after we do the Cauchy transformation, and we use the ACAT method to combine. And so this is a very, very quick calculation. It is quite accurate and super fast. So here is a result when applied to TOPMed lipid data. The discovery phase has 12,000 samples, the replication phase has 70,000 samples, and so here are the results for different masks. So it's nice to see that many well-known genes pop up. For example, PCSK9 and the NPC1, they are already drug developed for those two genes, and they are both loss functional variants and missense variants associated with LDL. And also, the SCAT also allow for the tissue-specific functional annotation. So as you can see that for lipid, we know that the liver will be relevant tissue. When one uses the liver epigenetic score as a weight, you can see that help with the more discoveries. And so computationally, when we do very large data, it has to be scalable. And so here is a computational speed. And so you can see that for using the STAAR for gene-centric analysis for 30,000 samples, it only take a half an hour. For 500,000 samples, take 1 hour if we have 100 CPUs. For genetic region sliding window approach for 30,000 sample is 15 hours, and then for 500,000 people, 25 hours. So it's quite computationally scalable because we use the score statistics and also sparse GRM to fit the linear or logistic mixed model. So takeaway from part one is FAVOR provide comprehensive whole genome variance functional annotation database and portal to to learn the functionality of any given variance and also annotate any whole genome sequencing studies. And also STAAR provide a powerful approach to empower real-variance association by dynamically incorporating multiple variance functional annotation. So let me move on to part two. So how can we do the meta-analysis by incorporating functional annotation? And so we propose this procedure called MetaSTAAR. So this is a joint work of the Xihong, Zilin, and also my former postdoc, Corbin Quick. The major challenge when we deal with the real-world analysis is we need to efficiently store the score statistic and covariance. And so when we want to do that across the genome, so how can we store it efficiently? When there are so many real variants, and because for real variance analysis we need to incorporate LD structure when calculate those test statistics such as SCAT. And so here I show you the storage and the time using the MetaSTAAR compared to rare metal. So as you can see that the compression time and for both the storage and also the computational time is much more scalable using MetaSTAAR, is a couple hundred more scalable compared to rare metal. So rare metal basically is not scalable for large biobank data. So how can we do that? So the basic idea is that we need to store the LD matrix or the covariance matrix using sparse matrices. So basically one calculates individual score statistics and then you store the individual score statistic. Then when calculates the covariance matrix, instead of storing the whole covariance matrix, you partition it into two parts and you save each part separately. And when you save each part separately, and then you save it using the sparse matrix. So for example, if one want to save this part, you can see that basically what we do is we create a banded matrix, and within the band, 500 KB band, and we only store the non-zero elements. And because of real variance and their many elements are zero, so we only store the non-zero elements. So that helps efficient storage. And if one compare the MetaSTAAR result with the pooled analysis, you can see that it has very good agreement. And basically the results are very similar to the pooled analysis. And the last part of the talk is how can we do those analysis in the cloud environment. In particular, cloud-based ecosystem is the trend of the field. And so in the traditional way, and we use the computing class, traditional computing, high performance computing cluster. So the ideas of high performance computing cluster is bring the data to the researcher. So for example, if one think about UK Biobank data, and the UK Biobank data, each researcher apply for UK Biobank data and store the UK Biobank data in the individual institutions research computing cluster. And for GWAS data, it's not bad because the GWAS data is not that large. But when one deal with the whole genome sequencing data, the storage is quite expensive. So this type of the data storage is not scalable. So there's no sharing of the infrastructure. Basically, data sharing is a data copy. So this is not scalable for large whole genome sequencing data. And so the BioData ecosystem, and so allow cloud-based data sharing. And so basically bring the researcher to the shared data. So by putting, for example, like TOPMed data or genome sequencing program data or the UK Biobank data in the cluster, and then everybody access that copy, single copy of the data. Of course, one has a backup copy, and one doesn't need to pay for the data storage and the individual investigator. So this is much efficient way for data sharing, and then one can develop a cloud-based statistical and machine learning method for analysis. So several cloud platforms such as Google Cloud or Amazon Cloud, and also NIH has in the last few years has been developing NIH data common, and for example NHGRI has Anvil. And NHLBI has BioData Catalyst, and so NCI has NCI Data Common, and by following this field principle. And so now, how can we do that for the whole genome sequencing analysis? And in the NIH data common, the idea is, suppose one has a raw VCF file, and then one can run the QC, for example, using TIL. And then that will generate the QC genotype data. And so one can convert the VCF file to the GDS file that will have much more scalability, and the size is much smaller. And then one use the backend database Favor and run this Favor annotator, this script, and for example, using the notebook, workflow, or BigQuery, and in Anvil, BioData Catalyst, or NCI Data Common, and annotate, say, UK Biobank whole genome sequencing data, then that will create the functionally annotated whole genome sequencing UK Biobank data, and then one run the STAAR pipelines and to analyze the whole genome sequencing data for association analysis. And so right now, the phenotype data can be downloaded from the DBGap, for example, like TOPMed data are available in DBGap. And so when this pipeline is established, and then one can run this in the cloud in BioData Catalyst for TOPMed, for example. So because we are currently developing this whole STAAR pipelines and Favor pipeline to allow investigator to analyze whole genome sequencing data in the NIH data common. So to take away, here are a few takeaways. So integrative analysis of whole genome sequencing data and the multi-omic data can empower discovery of real variance association using multifaceted variance function annotation, and also can help to study the causal pathway from the variance to mechanism and to disease. And so in both TOPMed and GSP, and so the both consortia and place a strong emphasis on diversity in whole genome sequencing study. And so this effort should be continued to prioritize greater diversity in genetic and multi-omics study to empower discovery and reduce health disparity in genetic studies. And also the cloud-based analytic form are the trend. So if we need to continue support developing scalable analytic tool and resources in the cloud, and also address the cost issue. I would like to thank many of my former and current lab members and who have contributed in many different ways through years and on the talk presented. Thank you.